Well, first of all, thank you very much for this kind of introduction and also the opportunity to speak here. I think already with the previous lecturers, it has become a time-honored tradition to emphasize how fantastic your achievement is at putting together such a nice conference already at the undergraduate level. And I would also like to chime in that, when we were at that age, of course, we didn't get anything done, and we were still busy trying to connect modems to the internet that was largely empty at the time. So I would like to encourage you to be interactive in the session. I think it was challenging for you, or daring of the organizers, to put the computational session so late in the day, just before the lunch. I hope you're going to be like hungry animals, attentive, and as we heard in the previous talk by Anna, hopefully we haven't slept much, and this is going to be a hard task on which all of your cognitive resources can be focused. So since I know this is quite a diverse audience and a young audience, I thought it may be a good idea to first of all define a little bit what computational neuroscience is. I mean, I think Matthew has already presented a little bit of computational work before, so you've already gotten a first glimpse. But I thought, just for everyone, especially the people that maybe work more on the cellular or let's say anatomical side or so, to say why is computational work interesting. I mean, of course, at the basic level, we could say, well, the brain is some sort of computer. And maybe some of you would take issue with this. I mean, of course, the brain is not a computer in the same sense like we have these computers here on the desk. But maybe we could at least agree that by natural selection, there has been certain principles that have been selected, sharpened through evolution, that now define and guide neural operation. And I think what computational methods can do is help us really get to the bottom of those principles, both in terms of analyzing them and also defining them, describing them in a manageable way. And so, while the zebrafish here, depicted here, is not going to play any role in my presentation, but I think, so we work on it as well in the lab, not on auditory processing. But the point is, this is one of the latest preps where you can record up to 100,000 or close to 100,000 neurons simultaneously. And you can probably imagine that in such a system, kind of your old intuitions fail, and you really need to use computational techniques in order to understand how that large-scale system is interacting. Let me just introduce what computational neuroscience and theoretical neuroscience has done by some of the greatest people in the field. So Jan Ebbett in their book Theoretical Neuroscience already defined or said theoretical neuroscience encourages crosstalk among these subdisciplines by constructing compact representations of what has been learned, building bridges between different levels of description, and identifying unifying concepts and principles. And I think it's important to realize that computational neuroscience is not just about making precise predictions. I mean, that's also important. But it's also important to try to distill huge amounts of data into manageable formulas. And I think, as we've seen in Matthew's talk, this is a nice example where a computational principle can guide you to define certain research questions in such complex data sets such as fMRI data sets. So what kind of levels exist in computational neuroscience? Well, I mean, I would say the top, in a certain sense, you have these pen and paper approaches. Yeah? So here, like the classic equation formula. These are top-level viewpoints where you try to summarize a huge amount of data into some principle. And often these approaches are normative, so you say, well, maybe some principle like efficiency or optimal performance should be maximized. And we try to see to what degree this kind of a principle is then realized in the neural responses. Then sort of at the other end, you have something I would call supercomputer analysis. So maybe you've heard of the Blue Brain project, which was the precursor of the Human Brain project. So these are projects where one tries to be extremely detailed in how one simulates the brain. And by providing this extreme level of detail, one hopes that eventually the resulting system is going to have properties that are shared or similar or can help us understand the properties of the real system. Because of course in such a simulation, if you set it up correctly, then you have full control. You can do any experiment you want in such a simulation, which enables you to make predictions. Now, there's also this middle ground that I would call the computation analysis, and that's in fact mostly where the realm where we are working, which is using computational methods to estimate certain things in a more detailed way than possible kind of with simpler methods. So for example, you can estimate multidimensional response kernels that try to characterize what a neuron is doing. You can have dynamical models that describes how a certain system or subsystem functions and processes the data. And these dynamical models are then often linked to principles that you will, that they embody. In the second part of the talk, I will show you one of those. Yeah, so these computational principles are then also expressed in kind of intermediate level formulas that are, I would say, sitting a little bit below this level of generality. So, since today I'm going to speak mostly about the auditory system, I thought I would give you a little bit of a flavor of what kind of computational principles have been identified in the auditory system, and I'll speak about some of them today. So on the lowest level here you have the cochlea where the sound is coming in and is first transformed into neural signals. And there, what I'm going to tell you today is about how the frequency transfer happens and what kind of sparse stimulus reconstruction tells us about why the representation is the way that we find it. Then the second level here, so here in the brainstem, you have synaptic integration. And what I'm going to try to show you is that there's a certain type of gain control implemented that helps you to remain very precise representations of the stimulus, even in the face of very complex or loud stimuli or large variations in loudness. As it, for example, happens when I sway my head left and right here. So then next, there's sound localization in the plane. So this is the thing that has been studied a lot in the auditory system, because as you maybe know, so in the brainstem, in the auditory brainstem, a lot of pre-processing is performed, much more than in most other sensory systems. And the reason for this is that the precision at which the sound localization has to be performed is extremely high, so that's sub-millisecond precision, and that you really need to do close to the periphery in order to lose that precision. Then next, if we look at the auditory cortex up here, then there's a principle or a model that has been put forth to try to explain these neural responses, which is called a modulation filter bank, which essentially is like a second order frequency transformation that expresses the sound in a particular way and is able to predict a lot of both basic response properties but also behavioral properties. So the so-called cortical model that was defined here by Chi has since been used in probably 10, 15 studies to different degrees of kind of perceptual level. It has been quite effective in accounting for human perception. And then here on the top sort of, the integration of complex sounds in decision making. So I'll say much more about this in a minute. But so the point here is that in complex scenarios, you need to perform something called evidence integration. So decision making isn't just detecting the beep or something, but it means that you have to be able to detect a complex change. And you're wondering how is this performed. So these are the three topics that I'm going to talk about today, each of them for like 10 minutes. Okay, so you of course all know this. So sound is entering the outer ear here, is then processed by the middle ear here, the tympanic membrane, and then enters the cochlea. And so the first time that this information is now transferred to neural signals is here in the auditory nerve. Yeah? So the auditory nerve is essentially a set of neurons and their axons that translates or transducts the transformed information from the cochlea then to the brain. And you can try to characterize the neurons in the auditory nerve by presenting this extremely simple stimulus. So it's just a click stimulus, meaning it's a very brief impulse that goes positive and then back to zero. And you can see what are the neurons doing of their response. So here's some examples, and here these frequencies indicate the dominant frequency with which they respond. So what you see here is essentially a modulation of the firing rate of those neurons. And you see that they are modulating the firing rate quite rapidly up and down. And you see that there's different fibers, and different fibers have different preferred frequencies. So if you think about the cochlea, you probably remember from your lectures, it's essentially extending a sound into a lot of different frequencies. And so if you measure the auditory nerves, you then find different neurons that, through this click, respond to different kind of intrinsic frequency of their response. You can now model this in a rather simple way. So I mean, you just define a function that accounts for this. So the so-called gamma tones are an example of this. So you have an exponential function that defines this envelope, you have a cosine that defines the oscillation inside, and then this first function which defines the decay over time. So that's all good. I mean, you can write a function down for it. Now you can first of all ask, well, why is it useful to know about this response property of the neurons when you perform some modeling? So here's a comparison of what you get if you perform a more standard short-term Fourier transform. So this essentially means you take the snippets of sinusoids that you use to transform the sound, or you perform the so-called cochleogram. So cochleogram means you take these gamma tone filters and you process the sound through it. So for example, here you have low frequencies and higher frequencies, etc. And you see certain things. I mean, you see that, for example, the cochleogram allows you to have lower, more detailed resolution at the sort of essentially expands the lower frequencies here. And it also allows you to be free of the choice of sampling rates. So sampling rate, essentially, in this case, you have to pre-choose. In the cochleogram, you pretty much stay in the same sampling rate that you started with, and you end up with a continuous signal of the same sampling rate. Now, what this allows you to do is, for example, to have improved predictions if you want to then model neurons that are further away. So the MMTB is a nucleus that's a little bit further down the processing chain. And if you want to model the responses of this nucleus, you're much better off using this as a kind of pre-processing step than using here the short-term Fourier transform. But so at this point you might say, well, that's all nice, that's kind of technical, but is there anything else you can learn from those shapes? And essentially ask, is there a more fundamental basis of why these filters look the way they do? And so there was a series of 2 studies, beautiful studies by Luigi and Smith and Luigi, both published in Nature in sort of the early 2000s. And what they did was to try to represent the sound. So here you have the sounds in gray in the back here, by an activation of individual filters. And so what's important here is what you see here already is the outcome of their estimate. So they did not put in any information about what these filters would look like. So they just put in, estimate any filter that is able to reconstruct the sound. So in the sensory reconstruction, that minimizes the residual, but in addition, make it in a way that is very sparse. So sparse here means to be very efficient, because the more you spike, the more energy you expedite, and so it's useful to be efficient on this level. And so the amazing thing is now that doing this approach, they were able to fit functions here in red, and if you then compare them, by matching them with the auditory nerve, you see that they obtained an extremely precise fit between the actual auditory nerve responses and their estimated kernels. And just to emphasize this again, so they did not put in any information about the cochlea, about sounds in the world or whatever. So it was just based on a sparse reconstruction principle. And here, there's sort of another pearl of the study is if you now look at the filters. So what you see here are the center frequencies, so the speed at which this modulates, and the bandwidth, so how wide it essentially is. And if you now in this space, so every filter here, whoops. So if you take one filter here, that essentially ends up as one point here. So it has one center frequency and one bandwidth. So every point here of these, these colorful dots here, these are filters, and the different colors now indicate with respect to what stimulus set they have been estimated. And you see, if you have vocalizations, then the vocalizations are kind of narrow band. If you have just environmental sounds, then the environmental sounds are, have a higher bandwidth. But if you compare the middle range here, so these are vocalizations together with environmental sounds, which arguably is actually what we are exposed to on a daily level, then you see that the estimated filters very nicely trace the underlying data set here, the blue dots, which are the real filters. So it's a very beautiful example of a principle, so kind of a normative principle, as they say, which is sparsity, together with this estimation principle, that lets you explain the way how certain parts of the nervous system, in this case the auditory system, are reacting in response to the natural statistics together with that principle. So I would like to continue this line, or bring you to the next higher level, namely the auditory brainstem. So that's where the auditory nerve fibers arrive. And show you how we continued this work, and then we're able to show that sparsity is actually a principle that is created by inhibitory influences in the brainstem. And so this is joint work with Christian Keine and Rudolf Klüpsam, and Christian Keine is really the one who did all the recordings for this. So before getting into this, let me just mention that there is a stark difference between stimuli that we encounter in the world and stimuli that we encounter in the lab. And if you're so kind to just listen to the first stimulus, I didn't test the audio by the way. So first of all, here's the Gilbert stimulus. And now the laboratory stimulus. Okay, so they're slightly different. And so, of course, some people have used more complex stimuli. I'm not going to try to say this. But the point is that the largest part of the characterization of the auditory periphery has been done using very simple stimuli. And what we tried to do in this study, in a similar way, like Luigi had done it by estimating against natural stimuli, is to go into a more general context. So, looking at contextual integration, and by contextual integration, I mean concurrent stimuli. Which are, in this case, for example, of course, you don't have just the tone at one point, but you have a wide frequency range that's being stimulated. And then the second part of the talk, stimulus statistics, so things that happen or develop over time and over frequency. So to give you the kind of place of action of where we did those recordings, let me just walk you through a little bit of the brainstem architecture. So sounds reach the ear, then are transformed by the cochlea, so we've already talked about this. Then here the sound goes through the ribbon synapse and then comes here to the spinal ganglion neuron. That's actually the same neuron that is sending the auditory nerve fibers. And the auditory nerve fibers here now arrive in the so-called cochlear nucleus. And we are recording here in the AVCN, the anterior cochlear nucleus. And in particular, we're recording from the so-called spherical bushy cells, SBCs here. These ones up here. And that information is then the most important, and I guess the only information, that really feeds into those sound localization centers. Yeah? So you have here so-called MSO and LSO. These are the two centers that enable you to localize sounds in the plane, so essentially to know whether a sound is coming from left or right, forward or backward. So we've been recording from the end bulbs of head, and so in particular the way that we do this is that the animal is anesthetized. A single electrode is approached to one of those connections, and then you're able to see both the pre- and post-synaptic side in one recording. So you're able to see the input and the output of the synapse, and then study the transmission under in vivo conditions. So this is called a juxtacellular recording. So it's essentially a glass electrode that is brought very close to this junction, and by being close to the pre- and the post-synaptic side, you can see both. So here's what this looks like. So you see the so-called pre-potential, PP here, which is the incoming action potential. Then you see the postsynaptic EPSP. And if there is a spike on the postsynaptic side, then you also see this large postsynaptic action potential. But so the red and the black here is now the difference between whether you have a transmission or you don't have a transmission. And of course, the important question is, why under certain conditions do you have a transmission and in other cases you don't have a transmission? So if you now take a look here at one of the tuning curves of those cells, then you see here that this is just the tuning curve of the pre-potential. So this is essentially what's delivered by the auditory nerve. If you compare this now to the output of it, then you see essentially two things. So you see that there is this inhibitory region here, but you also see that the output here is much, less strong. So this is shown on the same scale. And what's important is that previous research has largely focused on the question of, okay, so why do we have this this large inhibitory region here on the side? Is that maybe, for example, useful for sharpening the response in frequency? Whereas we tried to really kind of look at the elephant in the room, namely, why do you have this large-scale reduction that is actually completely overlapping with the excitatory input. So because if you, for example, if you here now look at which are the spikes that are not transmitted, and you see that the spikes that are not transmitted are essentially co-localized with the input. So you have a little bit non-co-localized at the high frequency region, but overall you have a high degree of co-localization between the excitatory and, okay, at this point, let's say the non-transmitted ones. So is this really inhibition? Was the next question we asked. So you can do this by applying blockers for certain inhibitory transmitters. So for example, we used here strychnine, which is a glycine antagonist, and that was locally provided in vivo in order to modulate the synaptic transmission. So again, we look at the same kind of paradigm. So we have the input and so on the presynaptic side, the response curve, and then on the other hand, the response curve on the strychnine. And you see that there is no change in the response properties. On the other hand, you also see this statistically across multiple cells. If we look on the output side, then the output side, you see that two things are happening. So a good part of the inhibitory region here is going away, and on the other hand, the rate here is increasing. So notice it's not increasing to the same level. So not all failures of transmission are due to inhibition, but a good fraction, like more than 50%, are really dominated by inhibitory inputs that is especially active under acoustic stimulation. Yeah, and so if you look at this, so the failures that we had before with essentially the region in which this was elevated, then these coincide. So now, what we can do computationally in more detail is to ask not only what is the frequency or the level and frequency dependence, but we can also ask what is the precise temporal dependence between the inhibitory input and the excitatory input. So we do this by using a randomized input stimulus. So that's essentially just a bunch of randomly placed tones, and then we use this technique called normalized reverse correlation to estimate the response properties of the neurons for both the pre, so the auditory nerve fiber, and postsynaptic side. This really bushy cells. And so if you do this, then you get here the presynaptic kernel and the postsynaptic kernel. And so on the individual cell level, you don't see too much. I mean, you see it's a little bit less, it's a little bit narrower in frequency. You have a little bit of a negative part here. If you average over many cells, you see that here, if you center it on the preferred frequency of the cell, you have an inhibitory action that is centered mostly on the preferred frequency of the cell, and then more or less symmetrically distributed around it, and integrates stimulus information over 10 milliseconds. So just to make this intuitively clear, so this you should consider like a template that you put on the stimulus spectrogram, and then you integrate essentially all the negative regions here with the energy in the stimulus spectrogram. And then this energy is what determines the inhibitory effect on your synaptic connection. So to put it in like a simpler case, so for example, if you have a stimulus that occurs for 10 milliseconds a little bit above, then that will, scaled by the amount of blue that we have here, influence the excitatory response and lead to more or less failures. One thing that's quite exciting to us is that this is actually very close to what you find in cortex. So this is a very good paper by Bernhard Englitz in 2003 in Nature, where what they were doing is to do in vivo intracellular recording, and they mapped the excitatory and inhibitory conductances. You can see that they're extremely well matched in frequency, so the acoustic simulation frequency, and also in timing. Yeah? So we don't see this here because it's 100 millisecond time scale, but there's, like we have it, kind of a small delay between excitation and inhibition, but you have overall a very good level of co-tuning between the two. Now, what are the functional consequences of this? So first of all, if you look at the rates, or the simple way of measuring this, for pure tones, then you see that the difference in the rate in the presynaptic side is quite substantial. On the postsynaptic side, it's still significant, but it's been much reduced. If you look at the same thing for natural stimuli, now here's something interesting. Namely, whereas you still have a very big difference in firing rate on the presynaptic side, on the postsynaptic side, the firing rate does not change whether you have that synapse or that neuron in silence or in loud stimulation. And so the inhibition essentially is, as we say later, is providing some gain control to reduce the activity to stay in a certain range, more or less independent of how loud you stimulate, and that's more effective in the case of natural stimuli. So we looked also at this property that Luigi looked at as well, namely the sparsity of the response of these Kabushi cells. You see here the presynaptic versus the postsynaptic sparsity, and you see that for essentially all cells that we tested, the sparsity increased here. And so whereas the estimate that Luigi performed was really for the auditory nerve fibers, what we can see here is that the sparsity even increases in the second order transition, which kind of chimes with their principle of increased efficiency for representing the auditory input. But in addition, also, if you look at the reproduction of the response, so reproduction here refers to if you stimulate the same thing twice, how well is the response in the first matched to the response in the second case. And you see that again, for essentially all cells, we find an increase in the reproducibility due to this inhibitory influence at the synapse. Now, again, continuing in a similar way how Luigi worked on this, we looked at how the reconstruction of stimuli is influenced by this. So we use an auditory stimulus, then look at the neural responses of the auditory nerve fibers, the EPSPs that didn't make it, and the outputs of the spherical Bushy cells. We then look at the short-term Fourier transform or this gamma-tone filter bank output of the stimulus, and we try to relate the two. So what we do is we estimate these reconstruction filters here that allow us to use the activity and predict what was the stimulus that this set of neurons was listening to. So if we do this, then we have here some reconstructions, and we can now compare how are the real to the reconstructed stimuli. So the mathematics is pretty similar to what they have, although we didn't use the sparsity as a criterion here for estimating the filters. So now what does this look like? So first of all, so here's the real stimulus. Then here we have the auditory nerve fiber-based reconstruction. And here you have the, so A is here the spherical Bushy cell reconstruction. And one thing you can see is that essentially you're much grayer here. So whereas here you have kind of these deep dips in the spectral energy of the stimulus, they are really well represented here, whereas here it's more grayed out. So a lot of this detail in how the modulation is encoded is essentially lost. And you can already guess by now why this is, because you have gain control that limits the range of the responses. And due to this limitation, your reconstruction is also more limited. So what does this mean now? Well, if you compute the correlation between your reconstructed stimulus and the real stimulus, you actually find that after the synapse, you're doing worse than before. So this is something that may come to a surprise. But then on the other hand, if you take a look at another measure, namely how is the temporal precision of the reconstruction, that is improved. So you have a trade-off here between temporal precision and whole level representation of the stimulus. So you lose whole level representation, but you gain in temporal precision. So to kind of put this together in a little cartoon, so if you have the stimulus here, which has some higher deviations, and then also some like large changes in level, and you compare it to the auditory nerve fiber representation of the stimulus, and then the spherical Bushy cell, then this is roughly what happens. Namely that a lot of the kind of off-baseline activity is subtracted out so that you end up with just the peaks in the output. And if you now look at just these peaks, and you look at multiple trials, then a lot of the try-to-try variability is taken out in the SVC. So that the SVC output now is sparser and more reproducible, which is kind of the key criterion for performing accurate sound localization. And then the way how you can then interpret the inhibition is namely that it's integrating over 10 milliseconds, so we saw this in this kernel, over some wide frequency range. And the functional value is that the response is more precisely timed and keeps it within bounds for these loud wideband stimuli. So, now this is on the brainstem level, but since we've seen so many cortical work today, I'm also happy that I can present a little bit of cortical level auditory principles. So this is on evidence integration of auditory textures. So another natural stimuli, I'll give you an example in a moment. And so this is joint work with many people from my time when I was still in Paris, and so e-book, but also for the EG recordings, Ursula Gorska, who's a PhD with me, but also at the So first of all, probably many of you don't know what a texture is. So I mean, textures were originally defined in the visual domain, so this would be like a patch of grass, or some kind of anything that looks stereotypic, for a large extent. And so visual textures have been studied a lot in the visual domain, but not much in the auditory domain. I mean, how can we define a texture? So a texture is essentially I would say a complex stimulus that's generated by elements that by themselves can be complex, but that they occur in a more or less random manner. So it can be completely random, or can be constrained by some statistics. But as a listener, you don't know exactly when these elements are occurring, but nonetheless, you are able to recognize them. So I will give you some examples here. So for example, rain, the elements are individual drops. And the statistics between these elements is independent. I mean, one drop does not constrain when another drop falls or when the sound comes. But the interesting thing is that despite all this variability in the sound, if you hear rain, you know it's rain. Yeah? So I can give you 10 samples of rain, and they all are totally different in their precise temporal structure, but you recognize them based on their statistics. Another example is, for example, fire. So here you have these crackles that occur at certain times. You also have some more low-frequency sounds. Again, that's largely independent statistics. Then scratching. So scratching would be a physical process. You have to take your fingers and you scratch here. Then you have kind of a stick and slip process between your fingernail and the surface, and that's going to generate a sound that again is it's at a given moment it's randomly composed, but you can recognize it instead. Then last, speech. Right, so speech is actually not a good example of a texture because there's a lot of kind of strict dependencies between the elements of speech that make it quite dependent in terms of its statistics. On the other hand, one of the most classical examples studied in auditory neuroscience, so-called cocktail party that you will also enjoy tonight, is where a lot of speakers are speaking together. Yeah, then you suddenly have, again, multiple sources that you can't really individually process, but that have a statistic that is not fully independent, but largely dependent from each other. And again, there you would need to be able to kind of recognize or predict the stimulus, at least at a statistical level, for example, in order to have a good conversation at a cocktail party. So let me just show you an example. There's some audio again. So this here is an audio of this genre. And yeah, I'd just like you to listen to it and kind of track your own perception of how do you identify things inside this stimulus, and at what point can you hear the cuckoo or so. I'm not sure how well you could hear this now. The point is, so roughly here, you see that there is a sound that corresponds to a, I guess in German you would say Kuckuck. I'm not sure, the English cuckoo. The point that I'm trying to make here with this example is there is a lot of things happening inside that frequency band. Yeah? And at some point, you're then able to detect that there is a change in that pattern. And then you're able to detect that there's a new stimulus that is occurring. And what I try to kind of highlight is that in this kind of a task, you have to model the statistics of the previous stimulus in order to make a prediction that at some point is violated by a stimulus that is more complex or louder or different in any way, so that you can detect a different kind of stimulus. And of course, in realistic situations, you didn't know in this case what you were going to hear, but still you were able to identify this as a change and relevant change. And so essentially, the task here is how do you separate expectable variability within such a texture from actual changes in statistics, and very importantly, how do you perform the separation dynamically? And so dynamically meaning, you have a stimulus that kind of goes on, and at some point you need to say there was a change, but you don't know when that change is going to occur. So there's some important work from McDermott and Simoncelli on this, who already came up with a kind of processing cascade to model this. So we tried to study this in the lab, both to study how can you model it, but also where does it really occur in the brain, by using a simplified version of a texture. So the texture is essentially just defined by its marginal statistics, and these are little tone pips. And at some point, a random point in time, there is a change that occurs in the stimulus. And the change in this case is just that there is a change in the probability with which these tones occur in a certain frequency. And then subjects were listening to this and had to make a free choice of when they say they detected the change. And after the change, after 2 seconds, the stimulus ended, and if they didn't detect it until then it was scored as a miss. At the same time, we were performing EEG recordings over the whole head. So here's a little bit of some data from the EEG recordings. So first of all, let's look at the onset of the stimulus. So this is not the textural change, but just the onset of the stimulus. So as you would expect, in the central electrodes, so around here, you find sort of a clear N1P2 complex, with then what's also typical, often sort of sustained negativity. And you can see that that did not depend on the amount of change that we introduce in these paradigms. So the 50%, 80% just means how much do you change the probability distribution. Then if you look at the time of the change, you also don't see a systematic change in the signal here. If you then look at the time relative to response, then you see that the effect that you can get is probably coming from motor cortex rather than from auditory cortex, but feeding into that potential. But you see it's coming after the response, so it can't really be related to the integration of the statistics. On the other hand, if you look at the parietal electrodes, right here, then we see that for the stimulus onset, there's not much of a response. So they're not really strongly involved in just detecting a simple onset. But then when you look at the time in particular linked to the response, then you see that we have quite a nice distribution of responses that differ essentially in the final height that they attain and in the slope at which they approach this. And this difference in slope here as a function of change size, that's really what you would predict in the case of evidence integration, namely because you need to integrate, if you say you integrate evidence at a constant rate, then you would expect that you have a different slope for different levels of evidence. Similarly, the size is changed for the different levels of change size. And on the other hand, interestingly, the change, as a function of change time, the size decreases as a function of time. So we view this as a reflection of the essentially threshold criterion that collapses over time because you know that eventually the trial is going to end. So in order to perform well, you over time maybe reduce your threshold, but this is a little bit more speculative. So now, having this data set, we were wondering, and also the corresponding behavioral data set, we were wondering how can we model this in a simple way. And so there at the beginning of my talk, we had the scale. So we're really at the top of this. So we have a very simple principle, first of all, in how we try to account for this integration. So the way I would do this is to look at the representation of the stimulus. So this is a little bit schematized here for the talk. So you see here, over time, we have these frequency bits, and in these frequency bits, there's these tones occurring at certain times. And then at some time, there's a change, and then you see here, for example, the likelihood of the number of tones is increasing. So we perform this by, in each bin, running a pair of estimators. So a fast estimator that is integrating the immediate history of the stimuli, and a slow estimator, which is integrating sort of the more recent history. So if you think about this, so the slow estimator is essentially trying to keep track on a rather slow level with what is the current level of statistics, whereas the fast estimator is trying to keep track of what has changed in the very recent history. Now then we combine these estimators across our frequency bands in the decision stage that is independent, meaning that if any of them crosses a threshold, then we say that there was a choice made for detecting a change in the frequency band. Now, what does this look like? So we have here the two estimators, so the fast one and the slow one. And you see the fast one is kind of making bigger excursions. But then when the change really happens, then that's when it really starts to ascend. Whereas the slow one is averaging more. And as you can see here, at the time when the change happens, it for a longer time stays unchanged. And so if you then plot the difference between the two, and you see that at this point is the first time when the threshold here is reached, and this would be considered as a decision for a change in statistics. So what we've done here is essentially generalize previous static evidence integration models. So if you know any of the choice probability literature, so essentially this is the same type of model that is used there, although they only use one estimator and not the difference between two estimators. What we like is that it has just 5 quite intelligible parameters that are not kind of randomly fit parameters, but these are parameters that really characterize the dynamics of this process. And based on the stimulus, what we do here is only look at marginal statistics, but it's very, very readily generalizable to other statistics. So we've already done this in a separate project where we try to produce a filter that can filter out textures in a kind of more computational approach. And now what we do, try to fit this to human psychophysics to see how well can this account for human behavior. So first of all, here are the hit rates of the performance of the subjects. And so the thick lines here with the dots on it, this is the model. And then on the other hand, you have the data behind it. Here the dashed line. And you can see that for the hit rates, we get a very nice fit between the two. For the miss rates, in a similar way, also the false alarm rate. So false alarm rate, we essentially compute always up to the point where the change is made, whether people have already made a decision up to that. And finally, also the reaction times, except for this annoying little peak here, we can fit quite well. So overall, we have a correlation coefficient of 0.9. Yeah, so you can see that such a simple principle, adapted to the stimulus, can directly account for the performance that subjects have in this textual detection test. One thing that we then asked is, well, is this now, can we now bring this into a more physiological context? In order to do this, we came back to this model, the so-called cortical model by Chi et al., which is a little bit closer to physiology. So what you have here is that you have these, so they're called the modulation filter bank. And so you have a certain scale and rate filters, where scale filters are across frequency and rate filters are across time. And then you end up with a representation of the stimulus. So this is just a subset of the overall model representation. And you can again then apply a decision stage to this one, and in fact end up with a very similar performance curve. So the principle of how it filters it is slightly different. It's essentially taking a rectified sinusoidal weighting into the past. But in the end, it's a similar type of processing that you do, namely you have kind of a longer time scale that you compare with a shorter time scale. Then, of course, an important question that one would like to ask is, so what does this really look like on the neuronal level? So this is ongoing research that we're doing together with our collaborators in Paris. So on the one hand, we do recordings in ferret auditory cortex, both parietal and frontal cortex, while they perform this behavior, to see which stage is it really realized. I mean, the prediction from our EEG recordings by source localization is that it should be kind of in the internal bank of the parietal cortex, but that we would like to really verify in the recordings in the animal. And so what we're going to do in my lab is to record the same type of stimulus in the mouse, where we're able to optogenetically identify the cell types. And so this way we could kind of bring it down to, is this more, for example, is the context of which texture you're listening to, is that carried by inhibitory neurons, which would be the prediction from predictive coding. I don't know if you know this theory. So in this case, that would be the direct prediction that inhibitory neurons are kind of carrying the stimulus context, which in this case would be the texture. Yeah, so I would like to conclude at this point. And what I've shown you is that many computational principles can be used to characterize auditory processing, like in this nice work from Ruiki, where the properties of the auditory nerve fiber responses could be very well matched to estimates of stimulus representation, in particular in the case of the combination between environmental and vocalization sounds. Then next, that essentially co-tuned inhibition is a mechanism in the auditory brainstem that allows you to provide gain control, and in this way enhance the sparsity, and then in this way sort of achieving this principle that was talked about by Wiki. And then finally, that a dynamic statistic integration model, or that the dual timescale model is able to perform this dynamic statistical integration in a way that is very closely matched with the performance of human subjects. And so let me just thank the people that have really worked on this. So Ursula has done a lot of the EEG work, and Eve has been doing a lot of the computational analysis together with me, and Christian Keine has done the recordings in the auditory brainstem, and there's a bunch of collaborators that contributed in several points, and of course, funders. And so I would like to thank you for your attention, and hope that we'll have a good lunch after this. Oh no, wait, after the session.
Thank you for the introduction. Now we have some time for questions.
Yeah, first of all, I find it deeply fascinating what you told about. I've got one question which is maybe a little bit off the main topic here, but there is a research showing that we've got top-down connections going as far as cortical nerve itself. And I was thinking, is it possible, and if yes, what would be the mechanism of modulating this very, very early auditory input, and how can it affect what happens then in the brainstem?
Yeah, so, I think it's an excellent question, and I mean, I agree with you that even down to the, probably even down to the cochlea, there is modulation. I mean, probably in a similar way how complexity increases, I assume that complexity of modulation decreases as you go down to the brainstem. I mean, one very simple level of modulation that you can always hear when your sinusoidal alarm on the phone is going off is that you have these little muscles between your ossicles, between the ossicles in the ear, and essentially they contract as a reaction to the loud sound. And so what they do is to prevent and protect you from damage to your ear. And so I think in a similar way you would have predictions that are graded as you go down. So I mean, like here you have some kind of prediction. I mean, you have an environmental prediction of how loud the sound is going to be and how much you have to downregulate your response. But of course, that you could also say you make over longer time scales.
And you told about loudness, and maybe we also have got we have predictions that are telling us which sounds are meaningful. Yeah.
Yeah. No, I mean, I agree. I mean, for example, the texture case. I think the kind of prediction you have to make has to be pretty general. Yeah? You essentially would have to predict a certain frequency of features that occur, but you cannot predict when exactly they will occur. So then the prediction would have to be on the feature detector level to say, unless you reach a certain level of frequency, then do not react. Yeah. So I think that is
kind of suspended prediction.
Yeah.
Waiting for the signal to
exactly.
reach the level.
Exactly. Yeah. I mean, so the principle that I've shown with the kind of two time-scale integrators can be directly applied to any feature level, where you just essentially do the same thing. You say, okay, so you kind of have a long time scale on which you accept certain changes, then you have a fast time scale that if it exceeds the short time scale on a certain amount, then you detect the deviation. So generalized is pretty easy.
Thank you.